Hello everyone. So this is the third part of our chapter, which is cell cycle and cell division. So in the last class we discussed about quiescent stage. So we are going to revise it again and we're going to start it with that only. So we discussed that some cells of the adult animals, they do not appear to exhibit the division, like this heart cells in our body, heart cells and many other cells divide occasionally only. They divide occasionally, and when they have lost the cell because of any injury, cell injury, or due to cell death, then that stage only they will divide. Otherwise they are not, they don't divide. Okay, so they divide occasionally only. So what happens at that stage, they will not divide further, so they have to exit the G1 phase. They have to exit the G1 phase, and they will enter a inactive stage, which we call as quiescent stage, so there are some cells which do not exhibit division. They will what do? They will exit the G1 phase. They will exit and will enter G0. Yeah, what is this happening? Okay, so there are some cells which will not exhibit the division, and what they will do, they will exit the G1 phase. And will enter to an inactive stage, which we call it G0 phase. Okay, G0 phase, it is called as the quiescent stage. Quiescent stage, okay, of the cell cycle. Next, what happens? The cell in this stage will remain metabolically active. It will remain metabolically active, but will not proliferate unless they are owned to do so. Means that unless there is depending on the requirement of the organism, like what kind of organism it is, and depending upon what are the needs of the organism, whether it's needing to be to do the division or not. So it depends. Okay. Next, in animals, mitotic cell division is only seen in the diploid somatic cells. So there are some exception, like where the haploid cell divide by mitosis. So for example, in the male honeybees, okay, the exception is male honeybees. Next, against this, the plants can show mitotic division in both haploid and diploid cell as well. So from your okay, this is for you will read it. Next, we have M phase. So M phase, most dramatic period is this phase. Okay, so it involves, I've already told you, M phase is the phase when the actual division takes place. What takes place? Actually divides. When it actually divides, so we can say the mitosis occurs. Okay, when the mitosis occurs. Okay, so this is that phase. So it is the most dramatic period of the cell cycle. And what happens? The number of chromosome in the parents and progeny will be the same. Okay, so one more thing is that I want to tell you. Which is a very important thing. M phase is the sorry, I forgot. In this there is equational division. Equational division. So what do you mean by equational division? Equational division, when we say equational division, so from the word you can learn that equational mean equal, means when the chromosome number of the parents and the progeny cells are equal, that type we will call it as the equational division. So it happens in the mitosis only, because if the parent is 2n, the parent cell is chromosome number is 2n, then it will be going to make the progeny of 2n and 2n. Okay, so it is equational division. But if we are talking about meiosis, then it is a reductional division. So why it is reductional? From the name it is very clear. Then in meiosis, when the parents chromosome number is 2n, the offspring will get n and n. So here you can see that there is reduction in the chromosome number. That's the reason why we call meiosis as the reductional division and mitosis, which is the M phase, as the equational division. Okay, so this was very important. Okay, so for now we have some four stages of karyokinesis. So in the last class I've told you that karyokinesis refers to the separation of daughter chromosomes. Okay, and in the karyokinesis we have four stages. What are these stages? Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Okay, telophase. Okay, let's now discuss about these stages. First, we'll start with our prophase. Prophase is the first phase of the karyokinesis. It is the division of karyokinesis. Okay, so first stage we have of the karyokinesis, which is prophase, and mitosis follows the S phase and G2 phase. Prophase phases of interphase. So in this, what will happen? Look, so in the S and G2 phase we saw that the new DNA molecule formed are not distinct, but they were intertwined. Okay, they were like difficult to separate. Intertwined is something which is difficult to separate. Okay, so in the S and the G2 phase, the new DNA chromosome were distinct and intertwined. Okay, so prophase is marked by what happens in the prophase. There will be initiation of condensation of chromosome, like the condensation of chromosomal material will get started from the prophase. So the chromosomal material becomes untangled. First we have to, like, these were the chromosomal material. First we have to untangle it, we have to untangle it, like we have to सुलझाना इसको, है ना. We have to like it's like a thread and it is tangled, so we have to untangle it. Okay, हमें इसे सुलझाना है. But first the chromosomal material will get untangled during the process of chromosomal condensation. What will happen? It will get untangled, and the chromosome which had been under, like we have discussed that try to recall it in which phase? In the S phase, synthesis phase of interphase. What we learned that in the animal cell there was the centromere undergone to duplication in the S phase. In the last class I've told you. So now it will begin to move towards the opposite poles of the cell, and then the completion of prophase is marked by these points. What are these points? These points are: chromosomal material condenses to form compact mitotic chromosome. Chromosomes are seen to be composed of two chromatids which are attached together at the centromere. Next, chromosome which had undergone duplication, I've already told you क्या होंगे वो, they will start moving towards the opposite poles of the cell. And each centromere then gets radiate out microtubule which we called as esters. Okay, and the two ester together with spindle fiber forms mitotic apparatus. Okay, next we have, this is very important to learn, that cells at the end of prophase, after the end of prophase, the cells when we see under the microscope will not show the Golgi complex, the ER, which is endoplasmic reticulum, nucleolus and nuclear envelope. This will get disappeared when we'll see under the microscope after the end of the prophase. Okay,